Hello and welcome to another Piper Pod. Today we're going to be starting a series of anatomy physiology. It should be about a 14-part series, and the reason we're going to do that is traditionally at the school we don't get a lot of time to focus on anatomy phys, and it really is the fundamentals of all knowledge when you start to look at pharmacology, pathophysiology. Having a good understanding of anatomy and physiology really helps in your further education with these other ones. So we're going to start today's session looking at the blood. Okay, so moving straight into it, the blood is pretty damn impressive, as you all know. It has multiple functions. The main ones are obviously listed in front, but you can go really deep into the rabbit hole to find out all the functions of blood. But the main thing it does is obviously it's the main medium, it's the main highway for distributing your gases, your oxygen, carbon dioxide throughout. It also rids the body, helps rid the body of metabolic waste, and it's the highway. It transports all the catalysts, all the enzymes, all the hormones for pretty much every reaction, non-neuro reaction within the body. On top of that, it plays a massive role in the regulation of body temperature. We'll learn a little bit more about that when we look at plasma. And as we've discussed throughout these series, it helps in the regulation of pH. Okay, obviously through osmotic drift, through the introduction of fluid regulation, and the transfer of ions. It's also responsible for regulating the amount of volume that you have. So through dehydration, it will draw in more fluid. When you overload it, it will push out more fluid. So it has a strong regulatory properties in taking care of the actual circulatory volume. And what we'll discuss a lot in the next presentation is its role the blood plays in dealing with infection. So when we look at the blood and we break it down to its absolute composition, most of the blood is made up of plasma. So they say about 55% of the blood, total blood volume, is plasma. Now obviously this varies depending on someone's hydration status, but generally speaking it's around that. What else we have inside the blood is formed elements, so a series of different cell types that make up the rest of the blood. Now majority of those cell types are the big heavy erythrocytes, or red blood cells, and they're generally around 45%, and that leaves essentially the buffy coat, which is made up of leukocytes and platelets, which only contribute to about 1% of the total blood that you find. So starting at the top, we'll look at blood plasma. Now plasma is mostly water, okay, hence why it's the medium. So it's the main highway. It transports everything throughout the body, and it holds a lot of our proteins within it. Now the proteins are mostly generated by the liver, and of the proteins that are found in plasma, the massive abundance of it will be albumin. Now obviously there's a whole bunch of other proteins within it, but majority you'll find will be the albumin itself. Now we've spoken a little bit about albumin in the past, but its main role in the blood is it becomes sort of like an osmotic buffer, an osmotic balance. Because albumin is such a large protein, it has a lot of osmotic draw. So it helps bind the water into the vascular system in order to maintain the volume within it. If it wasn't for these large osmotic values, or these large presence of albumin and obviously other elements like electrolytes, and the water would just flow through the cells, in between the cells, around the cells, into the cells sometimes, because it's got nothing, it's got no osmotic draw holding it into the vessel. So that's a role that albumin plays. But on top of that, as we've spoken in the past, it's obviously a massive contributor to your acid-base balance. Okay, albumin itself being a weak acid, it plays a strong role in the balancing of your cations, anions, and ultimately your acid base. But when you really look down into albumin, you'll find that it's Also the catalyst for a lot of chemical reactions as well. So you could spend all day talking about albumin, so I'll stop so we can get on to something else. The next thing that you'll find inside the plasma is globulins. Okay, now these essentially make up your antibodies. Now in your next presentation within the lymphatic system and immunity, we're going to talk about antibodies in depth. Okay, so just realize that they're found in the plasma, and you'll also find the ability to transport lipids as well. Last thing that is mostly found in plasma is your fibrinogen, which we'll talk about later when we're talking about clot formation. Now your plasma has a massive role. It's primarily designed in distribution of heat. Now the temperature of the blood is around 38 degrees. Now I know that sounds a little bit high, but it is higher than your normal body temperature. The reason is it has to be higher in order to pass the heat out through the blood into the body, which sits around 37. Now water's probably the greatest substance on the planet at retaining heat, hence why plasma plays the biggest role in the distribution and the control of that actual heat. On top of that, as mentioned, it's the highway. It gets rid of all your waste, carries all your nutrients and electrolytes. It's the transport medium for the respiratory gases, obviously in line with the erythrocytes, and it moves all hormones and enzymes throughout the body. Okay, it's the connection. So that is plasma. From plasma, we have to look at the formed elements itself. So we're looking at the different types of cells and cells fragments. The reason I define the differences is the formed elements, only the leukocytes are actually cells themselves. The erythrocytes are essentially a specialized, non-nuclear, no organelle-based cell. And platelets, well, they're just fragments. Okay. Now we'll look at each one of these in depth, and we'll look at how they're formed, what role they play within the body, and then hopefully by the end of it you'll have a pretty good understanding of the formed elements that make up our blood. So we'll start with the largest percentage-wise of all the formed elements, and that is our erythrocytes. So red blood cells they're quite unique in nature, realistically. Now their obviously main role is the transport medium for your gases. So it's made up of 97% hemoglobin. If you were to take the water aspect out of an erythrocyte, the actual protein chains, or what makes up an erythrocyte, is mostly hemoglobin. Now these things are beyond tiny. Okay, we're looking at fitting 5 million erythrocytes within a cubic millimeter. Okay, so that is absolutely mind-boggling numbers. It just gives you an understanding of how small they are. Now they have a biconcave discs, so the reason that's important is by having this biconcave disc, which means a concave section on both sides of the erythrocytes, it actually has what's known as a large surface area to volume ratio, which means it has the ability to hold more hemoglobin than if it was flat. It also allows for flexibility within the erythrocyte itself, and that's important so then the erythrocyte can travel down the capillaries. Remembering that some capillaries are only one erythrocyte cell thick. Okay, so they have the ability to mold and bend to fit through these capillaries as well. Now, looking at the main component that makes up erythrocytes, we have our hemoglobin. Now, hemoglobin is made up of 4 different subunits, or 4 different globules. Each one of those globules contains a heme molecule, and each heme molecule has an ion. Okay, and not an ion as an ion, but actually ion as in the element per subunit. Now it's this ion that's attached to the heme that has that reversible bond ability to oxygen. So each hemoglobin has 4 subunits of heme that contain for 1 ion each. So each hemoglobin molecule has the ability to carry 4 oxygen molecules. Now what most people get wrong is they think that the red blood cell, or the erythrocyte, contains just one hemoglobin. But that's not the case. Each erythrocyte contains about 250 million hemoglobins, which means each erythrocyte has the ability to reversibly bind oxygen of up to 1 billion oxygen molecules to it, which is pretty cool. Now, when we measure hemoglobin, it's in grams per deciliter. Now, there's a slight difference between the amount of hemoglobin that is normal in males compared to females, and it's mostly got to do with testosterone producing more erythrocytes to plasma ratio. Okay, and that's all it is. So whenever you see a measurement of hemoglobin, now it's different across countries, the main measurement for it will be grams per deciliter. Now, when we look at the formation of cells, you'll see this this process a lot. So when we talk about red blood cells, we're talking about erythropoiesis. Okay, now erythropoiesis is the process in which erythrocytes are made. Now, every blood cell starts with the same sort of stem cell. Okay, and that stem cell is called a hemocytoblast. Now, hemocytoblasts are found within the bone marrow. Okay, so the red part of the bone marrow, mostly on your large bones and up at your epithelial plates themselves, is where you'll find them. Now, that stem cell, okay, the hemocytoblast, has the ability to turn into any of your formed elements. And what controls that is a series of different hormones or different cytokines that will give it the instruction to make a particular cell on a needs basis. So focusing purely on the production of the red blood cells, so the erythrocytes, we need to understand what sort of things drive it. Now, in the event that we need more erythrocytes, the body has the ability to produce 2 million erythrocytes per second, okay, as needs sort of go up. Now, generally speaking, the balance between erythrocytes is controlled by the bone marrow. So just on a needs basis, red blood cells die and it produces more red blood cells through the bone marrow. But in the event that we have too much or too little red blood cells, then we increase the regulation with a protein that comes from the kidney, which is erythropoiesis. Okay, so this works on a negative feedback system in order to maintain homeostasis, but as long as you know that the balance is usually taken care of by the bones, and then on a needs basis, it either stops producing erythropoietin if we have too much, or increases the amount if we don't have enough. So when we get these recipes from is, as alluded to before, your red blood cells will die. Okay, now the reason that they die is because they're not true cells. Okay, so as they're being produced, they go through the process from the hemocytoblasts. They get produced in the bone marrow, and what they do is within its own cellular organelles, it will produce a large amount of hemoglobin within itself. It will use its body to take the iron, it will build the amino acids, and it will just build a lot of hemoglobin molecules. Okay, now once it has enough hemoglobin molecules, it will dump its nucleus and its organelles that it doesn't need. So when we have an immature red blood cells, it still has an internal ER. Now what that's for is that essentially helps transport amino acid chains within. So the reason it holds on to that is because it hasn't quite finished storing all of its hemoglobin on the outside. So once that's done, it will dump the rough ER, and then it becomes a true erythrocytic cell. Now, as a byproduct of being this specially designed cell without the ability to regulate itself, without the ability to repair itself, it has a lifespan. Okay? A lifespan in which it cannot reproduce itself or repair itself like other cells, because it doesn't have any DNA and it doesn't have any organelles for building amino acids. So it becomes a tool of the body. Now that tool is obviously going to break down over time. Okay, it's going to degrade and it's going to need to be destroyed. So what happens is the actual red blood cell will fall apart and the body needs to recycle this hemoglobin in order to produce more red blood cells. So it breaks up the red blood cells, mostly hemoglobin, it keeps the iron, it keeps the amino acids, and it gets rid of the bilirubin. Now, I know that was a lot to go through. So we've added a picture in order to help the understanding of the life cycle, if you will, of the erythrocyte. So starting right at the bottom with the production. Okay, we know it's produced within the bone marrow, but let's just say that you have a case of anemia. You've lost a little bit of blood, your body has registered that it needs more oxygen carrying capability, so your kidney will release your erythropoietin. Okay, that goes into the blood. It stimulates an increase in the production of the red blood cells within the bone marrow. So that means that the hemocytoblast now will focus on the production of red blood cells, and the other cells will just have to wait. Okay, once the new red blood cell is made, basically as I described before, it has a life of about 100 to 120 days. As it goes along, it begins to fall down and it will get picked up by your macrophages, which we'll learn about soon, when it gets to the liver and the spleen. From there, it will be destroyed. The body will keep your heme molecule and it will throw away your bilirubin and it will keep your iron, which adds to your ferritin levels of your body. The globulin, the free-flowing amino acids, they will ascend back into circulation and they'll be utilised by other cells to make different polymer chains. From there, as we mentioned in the past, the bilirubin is a waste product, so it gets metabolized down and it gets injected through the gallbladder into the digestive system and gets excreted with feces. Your digestive system obviously in the need of producing more red blood cells will absorb any amino acids, ions, vitamin B12 in order to produce more red blood cells. And this cycle just continues every day of your life. Every time a red blood cell dies, more are produced. Each time you lose blood, more are produced. And the body works as a true homeostasis in order to maintain that balance. Okay, so it is pretty damn cool, if you ask me. I think that's enough on red blood cells. I'm sure you all agree. So we'll move on to looking at our leukocytes itself. Now, leukocytes are white blood cells. Okay, they are responsible for the immunity. Now, I'm not going to talk a lot about the immunity aspects of the leukocytes because that sort of thing will be covered in the next presentations on immunity. Okay, but leukocytes are the only formed elements that are real cells. Okay, so they have a nucleus, they have organelles, and they have specialized function. Their role is to keep our bodies safe from everything that is a non-self cell or molecule. Okay, their range is generally around 4,000 to 11,000 cells per cubic millimeter. Okay, and obviously that increases as needs basis. Whenever you have an infection, you'll have over 11,000. Okay, so looking at the distribution of leukocytes itself, there are 5 leukocytes with specialized function within the body. Leukocytes themselves are broken up into 2 different groups. Okay, we have what's known as granulocytes and agranulocytes. And the reason they're called these is that when you look at the cells, you'll be able to see granules. Now these granules are like specialized pockets of catalysts that have different forms. Or you won't see any granules, hence the agranulocytes, and they will just have a large nucleus itself. Now, as far as percentage of white blood cells that we have within our body, the main white blood cell we have is your neutrophils, okay, taking a large chunk. Next, we move to the agranulocyte lymphocytes, then monocytes, eosinophils, and basophils. Now, the reason That's important to know is when you're looking at a white blood cell screen, you should also know what the normal values are and what's supposed to be higher within the body. Now, a little acronym that they throw out there for you to remember the order in which is most prevalent is never let monkeys eat bananas. Obviously standing for neutrophils, lymphocytes, monocytes, eosinophils, basophils, as you go down. Okay, but more importantly, let's break down what each one of these cells do. Okay, because obviously it's not the same thing. If every cell did the exact same thing, we'd only have one, and we'd just call it a leukocyte, and that's it. So each cell has a specialized function. So starting with the way that it's broken down and where they come from, is we need to look at the process called leukapheresis. So leukapheresis is obviously the production of white blood cells. Now, it's the same process as we went through with the erythrocytic growth, but I just want to have note to the first division that we have from the hemocytoblast. So a hemocytoblast will either become initially a myeloid cell or a lymphoid cell. Now, your lymphoid cell will just produce your lymphocytes. Okay, now we'll focus more on lymphocytes later on, but that's it. So once it becomes a lymphoid, it becomes a highly specialized lymphoid cell. From a myeloblast, we produce the other white blood cells. So there is an extra step. It goes from a myeloid into a myeloid blast, and then from there we produce the rest of the granules, the granulocytes, and your monocytes as well. Now, what controls this formation is a series of different cytokines and a colony stimulating factor is the main contributors to what produces most of your white blood cells. Now, that is obviously done on a needs basis. So looking at that needs basis, let's look at what each particular cell does. So starting with your neutrophils, largest percentage wise of your white blood cells, and definitely the main granulocyte that you have. Now, if you want to look at the picture of a neutrophil and you see those small little granules, that is why they're called granulocytes, because when you stain them, you see these little granules within it. Now, with a neutrophil, main cell that goes through phagocytosis for bacteria and some fungi, but mostly bacteria. Now, these are attracted by a certain chemical that the bacteria releases. So once it gets into an area, then it will move into it and kill that particular bacteria. And what it does is it engulfs it and it produces lysomes that destroy the bacterial cell. What we need to understand about leukocytes is leukocytes have the ability to move out of the bloodstream. Okay, some more than others. It's not just contained within the blood sea. So leukocytes can go through what's known as immediate movements, and it follows a chemoid tracks. Okay, now what that means is as it's drawn to an area of infection by the chemicals, the toxins that are released by it, or other stimulus, it has the ability to move through the cellular structure along these special chemical trails. Once it gets to the area, it's able to go through its phagocytosis. So remember, neutrophils associated with bacteria. The next one we look at of the granulocytes is eosinophils. Now, eosinophils will produce this very specialized enzyme, and the main thing that they do, they have the ability to destroy parasites. So eosinophils are associated with the destruction of worms. Now, obviously a worm, a parasite, helminths, they're too large. They're much larger than a cell, so they can't just go through normal engulfing of phagocytosis. So what eosinophils will do is they'll land on the surface of the actual worm and inject lysomes into that cellular structure of the parasite and it'll digest it. So many eosinophils will digest it. So if you have a worm infestation and you will take a blood test, you will find an increase of eosinophils. That's how it works. Last one, we look at basophils. Now, basophils are the last of the granulocytes, and their granules contain heparin and mostly histamine. So these are responsible for attracting other white blood cells. So they'll get to an area of need, they'll release the histamine, and that obviously will cause vasodilation within that area, which will attract more, which obviously will help clean up the mess. So your basophils are associated with increasing white blood cells. Now, when we look at histamines, obviously I know most of you know anaphylaxis, and most of you have heard of the term mast cell. Now, the difference between a basophil and a mast cell is a mast cell is associated with a profound allergic reaction. So if you have something that is allergy based, the mast cell will release a large amount of histamines and you'll get systemic vasodilation, or a much larger area of vasodilation. Your basophils play a role in this process along with your immunoglobulin E, but we're digressing. But basophils are more of a localized problem solver. So even in the event that it is not allergy based, your basophils can still play and produce histamines in order to attract more leukocytes to the area. Okay, so that's the role it plays. Now this is the least prevalent of all of the leukocytes, but if you have a highly allergic state, a constant chronic allergic state, you'll find that your basophils are higher in numbers. Okay, so think of basophils as the helper for the rest of the leukocytes. Okay, I think that's enough on the granulocytes, so we'll move to the agranulocytes, which most prevalent is your lymphocyte. Now these ones are your highly specialized cells. Okay, these are responsible for your absolute specific immunity. These things can turn into all sorts of very specialized cells. So they can turn into B cells, they can turn into T cells, and they can turn into natural killers. But I'm not going to start talking about that, or this presentation will go twice as long, because these guys will play the biggest role in next presentation. So just realize that that's what they do. Highly specialized structures, and they mostly live in the lymphatic tissue, hence lymphocytes. And the last one of the leukocytes is our monocytes. Now these ones here, these are pretty cool. Okay, these are the largest of the leukocytes, and they have this really cool U-shaped nucleus that you'll see. Now these are your absolute ass kickers. Okay, they are massive in your phagocytosis because they're so large. They can engulf a lot of things, and they have the ability to move into other areas of the body. Now when they are in the blood, they're non-active. They just flow around the blood as monocytes. As soon as they leave the blood, they evolve and they become macrophages. So they move into the tissue, they move surrounding areas. These ones do it the most than any other of the leukocytes, and they become the main defense for your organs. So you'll find macrophages around the base of your lungs, cleaning up all the mess. You'll find it in your organs. You'll find it in your actual tissues surrounding your muscle, and these macrophages will take down any invading body. So if you were to get a splinter and that splinter turns into an abscess or a pus that grows around it, chances are it is your macrophages that are taking care of it. And also a pretty cool side note, it is because of macrophages that you are actually able to sustain a tattoo long term. Okay, the macrophages will eat the ink and explode under the surface of the skin, staying there for life, standing it. This is what they do. Okay, so there's your leukocytes. Now I'm getting a little bit excited about macrophages, so we might need to move on. So that's the leukocytes that we have. Okay, now the last of all the formed bodies that we need to talk about, or the formed elements of the blood, is our platelets. Now platelets are not cells at all. They're not cells. Okay, they are the fragments of a cell. So a large, if you remember back to the picture, you'll see this very, very large cell called a megakaryocyte, and a platelet is the fragments of that. So megakaryocytes, once produced, they'll move to the outside of vessels, and they'll sit on top of the vessel, and they'll inject their tentacles, for lack of a better term, into the actual vessel itself, in between the cellular structure, and then those tentacles break off and they become your platelets. Now, your thrombopoietin is the actual regulator for this. So when you have a decrease in platelet numbers, you produce more thrombopoietin, which in turn will produce more megakaryocytes from the hemocytoblast, and you'll produce more platelets. Now, obviously, platelets play a massive role in clotting. Okay, they provide a temporary plug and encourage clotting to occur. But we'll look into that in more detail because it's a pretty cool process in turn. So when you get a cut, now I'm not talking about a massive one, we talk about that all the time, just talking about just a break within the actual vessel itself. 3 things occur in order to stop that bleeding. The first, vascular spasm, then platelet plug, and then finally we go through coagulation. So looking at each one of those in more detail, we have the first, vascular spasm, which means vasoconstriction of that blood vessel. What they want to do is they want to reduce the overall hydrostatic pressure around that vessel in order to reduce blood loss and also increase the ability of the platelet plug forming. Now, as soon as you cut into the vessel, the endothelial cells of that vessel will release a chemical which will cause vasoconstriction and attract more platelets. Other things help as well. So if somebody is in pain, okay, you have your catecholamine response, which will cause vasoconstriction as well. And you also have a sympathetic response in big cases when you have a loss of blood pressure, which in turn will also cause vasoconstriction. So other triggers help this process. But even in the event, the absence of pain or a sympathetic response for a small cut, the endothelial cells will still release a chemical starting this process. Now, when the platelet plugs form, the way that they react is, one, they're attracted by the destruction of the cells. But the cells themselves also have free-flowing collagen fibers that come out of the actual vascular wall. So normally, your platelets can pass by your vessels without sticking. They're not always sticky. But when you add platelets to collagen fibers, they become very sticky. And once they bind to it, they release their own chemical that enhances vascular spasm and attracts more platelets. It's actually a specialized, a type of serotonin that's released by the platelets in order to encourage more platelets to come and increase vascular spasm. Now, as more platelets come along, they bind with the collagen and they produce this big platelet plug. Now, once this plug is there, we need the binding aspect of all this to occur. So as soon as a platelet hits the collagen, it starts the process. It starts the clotting cascade internally. As soon as the vasculature is cut, it also starts the clotting cascade. Now, a thing to note is calcium plays a massive role within the clotting cascade. And once you go through all the different clotting factors, you'll get down to the ability to transfer prothrombin into thrombin. That is essentially what is being produced. Once we get to factor 10A, we finally have the ability, we finally have the catalyst to change prothrombin into thrombin. Now, what thrombin does is thrombin is the catalyst for changing fibrinogen into fibrin and factor 13 into factor 13A, which helps produce a cross-link fibrin. Now, the fibrinogen, if we remember, is floating around the plasma. It's inactive. Okay, and it only becomes active in the event that thrombin activates it. Now, from there, we create this awesome fibrinogen matrix, which collect erythrocytes, platelets, like a big plug over the top, causes the liquid aspect to turn into a gel, and that binds over the top of these platelets, like a big net. And from there, the erythrocytes dry out, gets absorbed by the body, and because of the stickiness of the platelet plug and the vasoconstriction. The sides of the vessels are pulled together and sealed with this cross-linked fibrogen net. Okay? And that is probably the most simplest version of the clotting cascade you're going to get. Okay, but still very cool to know, and as long as we get the understanding of what triggers what in order to produce a clot. So that is your platelets. Now to bring it back to a medical perspective, is we're going to quickly finish off with blood groups, because we talked about the different types of blood. So we've got the general anatomy and physiology, but now we'll put it into self. So your blood groups, when they talk about blood groups, they're mostly talking about the red blood cells, and everything we traditionally know about blood groups and compatibility is focusing on the red blood cells themselves. Now every red blood cell has antigens on their surface. Now an antigen is a self-identifier. Okay? So every single red blood cell has a different antigen or a self-identifier that is on its surface. Now each body has the ability to produce antibodies. Now antibody is a key or a trigger that identifies a specific antigen in order to start the process to destroy it. Okay, so it is against a particular antigen. Now when you produce an antibody, obviously as soon as that particular antigen comes into the system, it has the ability to destroy it. Okay, because it sees it as a non-self, so it destroys it. Now the interesting thing is, most of the time antibodies are produced upon exposure. But in the case of the anti-A and the anti-B antibody, a lot of antis, but in the case of those two, once you hit the age of about 2 months, your body naturally produces the one that you don't have. Okay, now I know it's a little bit weird, and we hope to get pictures to try to figure it out, but let's just say that you were type A blood, which means as soon as you reach 2 months of age, your body will start producing anti-B antibodies straight away without any exposure at all. Okay, very unusual. Okay, and that's the reason why we have this blood group, why we have to have these blood groupings. Now when we look at our rhesus antigen, okay, that's D antibody, then that only happens on exposure, meaning if you are a negative, you need to be exposed to a positive before you produce antibodies to it. Okay, and we'll talk a little bit more about it as we go along. Okay, but just realize that your body will naturally produce antibodies for non-self. Now when we look right into the actual antigens themselves, we have to realize that there's about 30 different varieties of antigens that can be found on a red blood cells. Now the main ones that we're interested in is the antigen A, B and D. So the ones associated with ABO and rhesus factor, so positive or negative. But other antigens that play a pretty strong role, especially in transplant or long-term blood therapy, is MSS, Duffy, Sells and Lewis. So you might read a little bit about those. They're other antigens that cause a reaction, but not a vigorous transfusion reaction like the ABO and positives. So in an emergency, we ignore the other ones. But if we have the choice, a lot of these scientists will actually look into those antigens as well. Now, when we look at the blood groups themselves, if you look at AB, what that means is A and B don't have any anti-A and they don't have any anti-B antibodies within their system. Okay, so if we were to introduce those two antibodies to type AB, then we'll obviously get cellular aggregation. Okay, now what that means is essentially the red blood cells are broken down, they become more sticky, and they form clumps together. Now these clumps block vessels, and you get hemolysis, which releases chemicals which attract platelets, and then all of a sudden you end up with large clots in areas and you end up dying. So when we look at A and B, it doesn't have any antibodies, obviously, because if it did have the anti-A, it would destroy itself. If it had the anti-B, it would destroy itself. So type AB doesn't contain any antibodies at all. So if we were to introduce it, obviously you'll get a problem. As opposed to O, that we know, it is not A, it's not B, it doesn't have any antigen, so it produces both antibodies itself. So it actually has anti-A and anti-B already in their system. Both of them already have it in their system. So if we introduce them, they obviously don't do anything because they can't attack the O groups. Okay, now the reason that is important to know is because it helps us understand the difference between component therapies and blood transfusion. Now, we need to obviously match blood that is not going to cause this cellular angulation. Okay, so it's not going to cause this binding, it's not going to cause this destruction. Okay, so as mentioned, type AB has both antigens, therefore has no antibodies. Type O doesn't have any antigen, so has both antibodies. Okay? And when we look at the rhesus factor, we just have to be aware that if you're a positive, you can get negative, but if you're a negative, you shouldn't get positive. Okay, because of the chance of re-exposure. And that's particularly important when we're dealing with pregnancy, hence the reason why we give anti-D shots and that sort of thing. So looking at the table of who can donate to whom, obviously we know that O group is what's known as the universal donor. But if we sit back and think about that for a second, I just told you that O has both antibodies, anti-A and anti-B. Meaning that if we look at it on face value, if we were to just put O blood into AB blood, or A or B blood, it would cause a problem. The reason it is known as a universal donor, because that term only refers to your red blood cells. So when you donate blood, they break it down into component therapy. So if you are giving pack cells, just red blood cells, then you give O, because essentially the plasma's been removed. When the plasma's been removed, your antibodies have been removed as well, therefore it's safe. If you're donating plasma and you're infusing plasma, O is not the choice. The most universal donor becomes AB, okay, because that doesn't have any anti-A or any anti-B, therefore it can give to all of them. So when you look at component therapy, depending on the component, we'll distinguish which one you should give. The chart in front of you is looking at red blood cells only. Now if you're lucky enough to use whole blood transfusion, and what you want to do is you want to match perfectly the blood groups. So if you are an A positive, you get an A positive blood, okay, because you are getting all the antibodies, the erythrocytes and the plasma in one. So I think that should do it. That is anatomy physiology of the blood as an overview. The first of the anatomy physiology series. The next one that we'll go through, as alluded to throughout the presentation, is we're going to look at the lymphatic system and immunity itself. So until next time, take care.